And our today's speaker is Dr. Nico Müller. And just so you know a bit more about Nico, Dr. Nico Müller is a philosopher at the University of Basel. He wrote his PhD thesis in animal ethics and published his book Kantianism for Animals in 2022 with Paul Gray Macmillan, then worked with animal communication researchers as part of a research ethics consultancy team. Since 2023, he's a PI within a national research program by the Swiss National Science Foundation called Advancing 3R, and his project focuses on the issue of phase-out planning for animal experimentation. And I suppose with that, Nico, welcome. It's really nice to have you here, and I'll give you the floor, and let's give Nico a warm round of applause.
Thanks, everybody, for taking the time. Again, I should say, sorry for not making it last time. That was embarrassing. But I'm happy that we're all here now, including myself. And I'm excited to talk to you about an issue I call Lane-Petter's pipeline and why decreasing animal research takes more than replacements. That national research program that Tero just mentioned, that was created by the Swiss government in 2023 with the express purpose of reducing the overall numbers associated with animal experimentation in Switzerland. These numbers have been stagnant since the mid-90s. Since 1996, we've been using sort of 600,000 animals per year in experimental situations in Switzerland, and that number really hasn't dropped since then. And the Swiss government is interested in finding strategies for bringing that number further down, not necessarily to zero. I mean, they haven't made a commitment to any specific reduction, but they're interested in their options. And this national research program really has that task to find strategies for that aim. Now, I'm just 1 out of 100 people, roughly, and mine is only 1 out of 27 or 28 research projects. But from a philosophical perspective, I hope that I can, by thinking about the issue from a philosophy of science, ethics, political philosophy standpoint, help to think outside the box about animal experimentation policy and hopefully move the debate forward, not just academically, but also politically. Now, this issue of Lane-Petter's pipeline, this really has to do with a specific difficulty that I think we face when we try to reduce the absolute numbers of animals harmed for research purposes, because science is really hard to plan. And it has something to do with the pipeline dynamic that I'm showing you in the graphic on the first slide that will reappear around the middle of the presentation. And I'll go into detail about what that is and how I think we might be able to tackle it. The presentation will have 3 main parts. The first being some background information about this call for a planned decrease of animal experimentation. This is a debate that is not Switzerland specific. This really has been a surging debate in Europe more generally and even worldwide. So I'll talk a little bit about that, and then I'll focus in on that problem of Lane-Petter's pipeline. And in the last part of my talk, the most tentative preliminary part, I will speculate about some potential policies that we could take beyond just replacement policies to hopefully bring the numbers down one day. So let me begin with the first point, some background. The idea of replacing animal experiments with other methods that are not reliant on using and harming full live sentient animals, that's a really old idea. In the 1950s, a book was published by 2 authors, 2 young biologists called Russell and Birch, and was called The Principles of Humane Experimental Technique. And that is where the 3 R's of replace, reduce, refine come from. The idea is they advocated you should, if you can, if your research question permits it, you should use some other model instead of an animal. That's the replacement part. Then reduction means you should reduce your sample size within your study, so only use as few animals as you can. And refine means take steps to reduce the harm that you inflict on the animals that you do use. But within these 3 R's of replace, reduce, refine, replace really is first and the most important privileged principle. And it's the principle that people have the most hope about, that it can make science more animal friendly over time and get us towards lower numbers as well. Now, since this 3 R's principle and the replacement principle particularly was put forward, a lot of advances have been made, for instance, in cell culture approaches. So some animals are no longer used in science because, well, and also in industry because we can replace them with cell cultures. Other examples of replacements would be organoids. So that's structures, 3D structures made out of real human cells that start to behave and form structures a little bit like human organs. So there's liver organoids, heart organoids, brain organoids. These can be used as models in certain contexts. And another example would be organ-on-chip technology. These are small chips, usually made out of plastic, where real human cells are combined via minuscule capsules and channels so that they interact a little bit like they do in the body. Again, this can help us to model certain systems of the body. So replacement really has made advances, not in a way that has brought the overall numbers down necessarily, but there's certainly more replacements available today than there used to be. And there are a lot of centers and platforms across the globe that really promote this idea of the 3Rs and replacement in particular. Traditionally, the ambition behind replacement wasn't to replace all of animal experimentation or even a significant part of it, but only, some animal experiments, the ones where it's opportune. But in recent years, in the last 15 years or so, this ambition has become a little stronger. So we've seen political calls for a full phase out, full replacement, it's sometimes called. So for instance, I think the most important example would be in the EU's Animal Experimentation Directive, which is called 2010/63 EU, where it says in Article 10 that this directive represents an important step towards achieving the final goal of full replacement of procedures on live animals for scientific and educational purposes as soon as it is scientifically possible to do so. So here we have a major political body endorsing this idea that we might one day overcome all of animal experimentation, not just some, by way of replacement. Full replacement is the idea. And this isn't just a theoretical possibility, but it says it's a final goal. So it's something we should try to achieve. Now, this was welcomed by animal advocates and sympathetic policymakers, but it also soon led to frustrations because people started to feel that this was, more or less just a commitment on paper and that not enough was being done to actually move towards that final goal. And this led, among other things, to some European citizens' initiatives, one of them called Safe Cruelty-Free Cosmetics. An earlier one was called Stop Vivisection. Both of them essentially made the same point. We have the goal in Article 10. Now we need a strategy. And this is where all this talk about plans and strategies, oftentimes phase-out strategies, comes from. That's where the debate really started. The European Parliament itself has adopted a resolution that made the same point. We have the goal, now we need a strategy, which is the one I'm showing you now. What's important to know, however, is not every resolution of the European Parliament is a legislative act. This is a non-legislative resolution. This encourages the European Commission to act, but it doesn't force it to act. And the European Commission really did not do much about this based on this resolution. There's other contexts in which the call for a planned decrease and phase out was made. In the UK, there was a petition to the government. In Switzerland, we've had a petition to our parliament. And as we speak, Parliament in Switzerland is debating whether the country should adopt a phase out plan. So it's really a hot topic where I come from. The Dutch have been debating this issue for a while. In 2016, an important report was published by what's called the NCAD committee, which really laid out a kind of very rough phase-out strategy that the country could use if it wanted. Then subsequently, it didn't really follow that strategy. And now they are still interested, although they haven't really followed through with the plan. They are still interested in dealing with this. And if you want, you can right now participate in a participatory process by the Dutch government to give input on how they should take the next steps to transition towards non-animal research. So that's a hot topic there as well. And finally, I want to mention Germany, because the German government is, I believe, the only government in the world that has officially committed to developing a reduction plan for animal experimentation. So this was already in what's called the coalition contract. So the three governing parties in 2021 already communicated that they wanted to come up with a reduction plan for animal experimentation. Then for the longest time, you couldn't hear or see anything about this. But then last autumn, they did start a participatory process to get stakeholder and expert input to start figuring out what this strategy could look like. Now, details about this strategy are not public yet. We don't know how ambitious it's going to be or what steps it's going to include, but it, I think, does represent a paradigm shift in animal experimentation policy to even look at animal experimentation policy as something that calls for strategies, for transformative strategies. Traditionally, we look at animal experimentation as something to be licensed and regulated, not necessarily as something that we should strategically move away from. So I think this is really an interesting development. Now, this is a political discussion. And in political discussions, you're not forced to define clearly what you mean by the words that you use. Everything is up for negotiation. So there's a danger that everybody talks about phase-out planning or reduction planning, but people don't mean the same thing. So for a publication early last year in the journal called Altex, Alternative to Animal Experimentation, I looked at different proposals and political calls for phase-out planning, just to look if they operate with a similar notion of what it would mean to have a plan. And of course, there's big strategic differences, right? If you look at what PETA says about how you should phase out animal experimentation, they'll suggest very different measures from, let's say, the American EPA. So different players have very different strategic ideas, but I think still they have roughly the same notion of what it would mean to have a plan. And we can sort of distill that out of these different approaches. To have a phase out or reduction plan would mean to have a document that defines three things that all begin with the letter M, so they're easy to remember. The first is a phase-out plan or a reduction plan has to define measures. Who should do what? So it's an action plan, right? For a specific competent authority, for a specific institution. And these measures can really be anything that an institution or competent authority is able to do. It can have to do with revising regulations. People, when I talk to them about phase-out planning, usually they think I'm just talking about capping animal experimentation. So, Switzerland uses about 600,000 animals a year in experimentation. So they think I want to sort of put a quota on the licenses that we give out so that this year it's going to cover 600,000 animals, next year just 500,000 and so on until we reach zero. That's one idea for a potential measure you could take. It's particularly prohibitive and negative. It doesn't do anything for other approaches. So it's probably not a very good idea. It's just one out of a million different ideas you could pursue. You can also think about moving money around, right? Providing research funding, providing communities with funding so they can thrive, research communities, think about infrastructure, also think about institutions and advisory bodies, working groups, and so on. So this can be quite positive and it can be quite varied also. It doesn't have to be so prohibitive as people sometimes assume. The second thing you need is milestones. You need to be clear on what you want to achieve in terms of outcomes by means of your measures. These eventually have to be reduction goals towards the end of the plan if this is a reduction plan. But it's important to emphasize once again, the milestones don't just have to be about reductions in total numbers. They can be defined in any way you want. Anything that a competent authority or institution can hope to achieve can be a potential milestone. So for instance, one potential milestone could be so Finland aims to have an organoid facility in every major university by 2035. That could be a kind of milestone, right, on the way towards the eventual reduction of animal experimentation. What's important is you have the intermediate goals predefined. You know what way you're going to try and achieve a reduction in animal experimentation. And the third thing you need is metrics. You need to be able to tell whether you're making progress or not. And you need to be clear on what numbers will be relevant for that. Again, at some point, the total numbers that you have in your national animal experimentation statistic will become relevant, but they have by no means to be the only numbers you look at. It can be much more complicated as well. And then the idea is that these three M's of measures, milestones, metrics interact with each other. Right? A phase-out plan or reduction plan is not meant to be a prognosis that's set in stone and then you have to go and enact it. And if it doesn't work, you lost. That's not the idea. The idea is if our metrics tell us that we are missing our milestones, then we adjust something about our measures. Right? We make course corrections. That's what's strategic about this strategic plan. It allows for course corrections, which means that phase-out planning or reduction planning has to be an iterative process. It has to be a learning process as well. And course corrections along the way are part of the idea. That's a feature. That's not a bug. But of course, if we want to imagine such phase out or reduction plans, realistically, we have to think about what measures we are envisioning here. So what kinds of measures would be needed to decrease overall animal experimentation? Now here, a lot of different distinctions would have to be made. Animal experimentation is a huge field. Animals are used in testing and research context in very many different ways and for many different purposes. And I can't go into every one of the distinctions that would be relevant, but I'll just stick to one extremely fundamental one, which is the difference between regulatory animal testing and animal research, so for scientific research purposes. This is a distinction that the European Commission highlighted in its response to the European Citizens Initiative, Safe Cruelty-Free Cosmetics. And they were actually quite open toward the idea of planning a reduction of regulatory animal testing. They just were very, very hesitant to commit to a reduction of animal research. So what are we talking about here? Here you can see a graphic that shows the different purpose or some of the different purposes for which animals are used in the EU for scientific purposes. And I'll just talk about the three major groups here, going from 12 o'clock clockwise. You can see here that some 37%, sort of salmon colored, 37% of animal uses in that particular year served basic research purposes. After that came some 35% of animal uses for translational and applied research purposes. So overall, more than 70% of animal uses have some research purpose in mind. And after that, the last big group of animal use purposes are regulatory uses in sort of mint green at 6 o'clock. And this includes, for instance, chemical toxicity testing that the regulator asks for. So if you're a chemicals company, you want to bring a new chemical on the market, then the regulator demands certain data from you. You have to document whether your new compound irritates the skin, the eyes, if it causes birth defects, if it's toxic to the environment in different ways. And very often animal testing is used to supply that data. Now, as I said, the European Commission is quite open toward the idea of phasing out, or at least reducing and eventually phasing out animal testing for regulatory purposes. And they even wrote that the Commission will immediately launch the work to develop a roadmap that will outline milestones and specific actions to be implemented in the short to longer term to reduce animal testing. And that would be prerequisites for a transition towards an animal-free regulatory system under relevant pieces of chemical legislation. It seems that this is not just a commitment on paper. They are serious about this. They have started the participatory process to start working on this reduction plan. In the same document, they very clearly say no to doing the same thing for animal research in a scientific context. And the difference really is that in regulatory applications of animal experiments, the regulator has clearly defined what questions you have to ask, what data you have to supply, and what methods you can use to supply that data. So everything is well defined here, right? The regulator is very, very clear about what you need to supply. So in essence, you can draw up a full list, that's a finite list of animal experiments that the regulator is asking for still, that don't yet have an approved alternative next to them. And then you can go through the list and try to innovate and validate an alternative for each of those remaining animal tests. And you can go through the list that way, right? In some cases, you can also redefine the data requirements on the regulator's side. So maybe there are alternatives, but they give you data in a slightly different structure. Then maybe the regulator just has to ask for the data in a different way, and then alternatives become applicable. So all of this is very complicated. Scientific innovation is difficult. Regulatory innovation is often difficult, but at least the task is well defined. We know what to do in order to reduce and eventually phase out animal testing in a regulatory context. The same is not true in science. In science, the regulator doesn't tell you what questions you have to ask, what data you have to supply, and what methods are appropriate to supply the data. Science is much more open. And that's part of the DNA of science, that it asks new questions, approaches them in a different way, answers old questions with new data. And it's this openness of scientific research that really makes it so difficult to plan a decrease of animal experimentation. Because there isn't a finite list of the animal experiments we would have to replace in order to eventually get done or even make a dent in the overall numbers. And this is where the problem of Lane-Peters pipeline really comes in. It has to do with this open-endedness of science. In 1961, British scientist and regulator William Lane Petter published a book about animal experimentation, and he described animal research specifically as a moving pipeline. So it's not a finite list, it's a moving pipeline. At any given point in time, animals are being used in, a certain number of animals is being used in research, and a certain number of procedures is being carried out on them, and that's what happens inside the pipeline. Those are the numbers that you would see in a national animal experimentation statistic. But what you maybe can't see in a national animal experimentation statistic is the change in lineup that goes on all the time. So year on year, even if the numbers are roughly the same as they are in Switzerland, it's not the same animals being used. It's not the same procedures being carried out. There's a constant change of lineup. On the left-hand side of this graphic, you can see the innovation and uptake of replacement. So people come up with new cell cultures, new organoids, new organ-on-chip technologies, what have you, making some animals obsolete for research purposes, and these animals, so to speak, get to leave the pipeline. But on the other hand, on the entry side of Lane Petter's pipeline, there's also the innovation and uptake of new animal models. So people start asking new scientific questions, and they address some of them with new animal models or new applications of known animal models. So constantly, animals are also entering Lane Petter's pipeline. And the number of animals in the pipeline is determined by what happens at both the exit side and the entry side. Traditionally, we are focusing on the exit side, right? We have all these 3R centers and platforms. We invest in replacements, but we are constantly catching up to a scientific status quo that happens inside the pipeline. And meanwhile, new animals are entering on the other side. If the policy goal is to reduce the number of animals in the pipeline, or the number of procedures carried out on them, or at least the procedures that harm animals, then we need to be interested also in what happens at the entry side. And that's sort of a blind spot of traditional animal experimentation policy. So I want to think a little bit about what happens there and what potentially policy could do to slow down the entry rate of new animals into Lane Petter's pipeline without, ideally, too much of a cost to science. I want to argue that room for innovation in animal models is essentially infinitely large. So we can keep innovating new animal models at the entry side of Lane Petter's pipeline if we want. If policy doesn't intervene, this can go on forever. And one way to bring out this point is with a couple of examples, and I want to structure them using this theoretical model that philosophers of science sometimes use to think about animal models. It's simpler than it looks. It's essentially a model for how animal models work in a scientific context, what animals do in the lab. Up top on the left-hand side, I think you can see my mouse. Up top on the left-hand side, you can see the model. Those are the particular animals we're using, let's say 10 mice in our lab, and they are meant to be models for something else, right? They denote or represent something, and that's our target. If we're doing translational research, then the target would be some group of humans. If we're doing basic research, then maybe it's sort of a larger biological grouping to which the mice themselves belong. Maybe the mice stand in for all vertebrates or all mice or what have you. So both are possible, but they stand for something more than just these 10 particular mice. How do they achieve this representational function? Well, it's quite simple, really. They exemplify certain properties that are connected to properties of the target. So for instance, maybe our 10 mice have certain genetic pathways that are connected to the genetic pathways that our human patients have that we're interested in studying. So their genes are the same, or they interact in similar ways. Or maybe they share some developmental traits. A real life example would be that the heart develops in similar stages in humans and in mice. So oftentimes mice are used, mouse embryos are used to study cardiac development in an experimental setting. Now, this connection between properties of the model and the target is not just obvious, right? Animals are unlike humans in many different ways. What counts is that the particular properties we're interested in are connected in the right way. And this connection is established by what's called the key or the key principles. And this can get very complicated, but maybe the easiest way to understand it is to think of biological conservation, right? Mice and humans share a lot of evolutionary history, so some structures are conserved across evolution. Of course, there can be evidence, pro and con, right? This isn't just a dogma. This can be established or disproven. But the idea is, regarding certain properties, these connections hold. And that's how animal models work, essentially. They represent their target by exemplifying certain properties that are connected to properties of the target. And this can help us to think about where we could, if we wanted, innovate new animal models. I endorse none of these ideas, but they are theoretical possibilities. So one possibility is to use non-standard species. You can use a greater range of animals as models than is traditionally done. Some philosophers of science indeed worry that we are missing out on scientific advances because we focus too much on mice and rats and zebrafish, the most used animal models, and not enough on Antarctic ice fish or dune mice. But any animal could still turn out to have certain properties that are interestingly connected to properties of some target we want to study. A real-life example would be the use of non-human primates to study rare human diseases. The more we learn about gene variations in non-human primates, the more it turns out that they have some similar gene variations as have patients in, human patients who suffer from rare human diseases. So they turn out to be adequate models for something we hadn't even considered. That's sort of a low-tech way to innovate new animal models, if you want. Some people are very excited about this. Another avenue for innovation is to make new properties tractable. People have, thanks to gene modification, they've made transparent adult zebrafish. Usually zebrafish are only transparent when they're very little. People have figured out ways to make them transparent even when they're adult. So you can observe what happens on the inside, and that makes new properties tractable or more easily tractable. The same has been done with frogs. And to give another example, you can also use proteins that glow in the dark, fluorescent proteins, to observe what happens within cells of let's say, zebrafish. So you can track certain properties that would otherwise have been very difficult to study. So that's another way to come up with new animal models. You can also improve model target fit. Of course, oftentimes that's the reason why genetic modification is used in animal research. Maybe mice don't get the kind of cancer that you'd need them to have in order for them to serve as a model for your target. Sometimes you can make them have that kind of cancer. You can also sometimes improve the model target fit sort of by going in the reverse. So you go from, you study something that you already know works in humans by translating it back to an animal model and try to sort of enhance the zoom factor that way. You can also address new properties in your target. If your understanding of the target has evolved over time, then you can develop new animal models to address new questions. Let's say, for instance, if you have you hypothesize that the human immune system has certain relations to the gut microbiome, and you start asking new questions, and then you can, then you develop a new animal model. Let's say a germ-free mouse, a mouse that doesn't have any bacteria in its own gut, and then you give the mouse some strains of human gut bacteria and see how the immune system reacts in certain ways. So that's another way to develop new animal models. And last but not least, you can expand or refine the target population. Historically, it's been a concern that many animal studies used exclusively male samples of mice, and it turns out, or of animals more generally, and it turns out when you do that, oftentimes your studies are more relevant to men's bodies than to women's bodies, and that's, of course, a justice issue, and you can also take this as a prompt to design new animal studies that are either adequately mixed in their sample or even, to develop an animal model that better represents women. You can even go extreme on this. You can develop new animal models just for one particular patient. People do this in what's called patient-derived xenografts. When you take a cancer cell from one particular patient and implant it into one animal model, then you get a model just for that particular patient. That, of course, opens up a lot of opportunities in the area of personalized medicine, if you want, right? My point isn't that we should do any of these things. My point is that we can, if we want. And these different avenues for innovation within animal research, they cut across each other. And each of them in itself already gives myriad new possibilities. And if they cut across each other, I think the room for innovation in animal models just becomes infinite, for practical purposes, infinitely large. Meaning we can go on doing this as long as we want. If you come up with a new model, chances are there will be some questions, some scientific questions you can only address using that particular model. So we can reproduce animal research and the perceived necessity to rely on it forever, if we want. Now, the same thing that I've been saying now about animal models would be true about any other kind of model as well, about cell cultures, about organoids, about organ-on-chip technology. You can always use a greater range of models, make new properties tractable, improve model target fit, address new properties, and expand or refine the target population. For example, a traditional concern about alternatives to animal experimentation is that they cannot adequately represent a full organism, right? An organ on chip is not a full organism. People have the concern that this means they can't properly represent full organisms like the human body. But that's really just a concern about connection, right? So you can try to take this as a prompt to improve model target fit. Maybe you can hook up different organs on chip to each other and achieve what's sometimes called a human on chip model that more adequately represents full organisms in certain ways. So you can always try to improve on what you already have. Innovation really is open in any model. The question now is, what determines whether scientists focus on innovating new animal models, keeping Lane-Petter's pipeline going, or if they move towards more innovation in non-animal models, keeping scientific progress going without feeding Lane-Petter's pipeline. And that's where I get to the speculative part of my, or the most speculative part of my talk now. Because I think policy might be able to do something at the entry side without holding science back too much. The question is, what determines model innovation? And I'm not aware of any literature, despite looking a lot. I can't find any empirical literature that directly addresses this question. So what determines whether a researcher innovates a new animal model or a new non-animal model? But I mean, I think we can make reasonable assumptions. And my default assumption would be, well, researchers try to innovate the new best model for a given scientific purpose. The reason why people keep innovating new animal models is that they think that the animal model they envision will be the new best model for a given scientific purpose, for some question that interests researchers. So that would be my default assumption. You know, there's a scientific reason for model innovation. But if we go along with this assumption, there's two important follow-up questions. Namely, what makes a good model and how do researchers select scientific purposes? Right? Both of these will be crucial to understand where model innovation goes and what maybe we can do from a policy side to steer it in a certain direction. So I want to comment on these two questions in the rest of my talk, and then we'll go towards the Q&A. What makes a good model? There is good literature about this in the philosophy of science. In one paper in particular, I find very, very helpful to think about this, how to choose your research organism. This is written by 5 philosophers of science who've looked at how animal researchers actually justify their choice of animal in their publications. And they find that the justifications are in some ways scientific, but they are also very practical in some ways. And they argue this is actually fine because the best model for you as a researcher is the one that helps you make your best scientific contribution. This depends on certain scientific considerations, right? The model has to help you study the thing you're interested in. It has to respond to experimental interventions. It has to make properties tractable. It has to be durable enough so you can actually observe the behavior you're interested in, and so on. But practical concerns are also very important. You can't use a model if you don't have access to it, if you can't afford it, or if you're not trained to use it. So the best model for you will have to pass by some scientific and some practical criteria. And overall, they come up with a list of 20 different criteria, and I can't go through them all in detail now, but it's a really interesting mix between these more scientific and these more practical considerations. And all of the ones that I've just mentioned are on this list as well. I'm struck when I look at this list at how many of these criteria for what makes a good model for a given scientific purpose, how much depends on the environment, the built environment in which scientific research takes place. Most of these, maybe not most, but roughly half of these criteria are really not just about the model itself. They are about the relation that the model has to an environment of scientific research. At least the ones I'm highlighting in yellow right now are at least partially about the environment. Ease of supply. Can I just go online and does my institution have a form where I can just order the model? Or do I have to, figure out everything for myself? Is it very hard to supply a specific model? That makes a difference. how well I can work with a model. So ease of supply is one example. Standardization. Is there common vocabulary? Is there common best practice guidelines and protocols for how to use the model? Are there common reporting standards in the community? That's a matter of what work the community has invested into establishing those kinds of standards. So it's, again, environmental. Availability of methods and techniques has to do with devices and infrastructure that I might need. It's, again, a matter of the priorities that my institution sets. Epistemic resources, training requirements, informational resources. Those are a matter of documentation and education. Institutional support, financial considerations, community support. Those are about the priorities of the institution, the community, and the funders. And finally, even commercial and other applications are, to an extent, environment dependent, because if you've got a technology transfer office at your institution that already has experience in marketing the kind of model you're developing, then that's an advantage, of course. So again, to an extent, this is environmentally dependent. So what makes a good model is not just a scientific question regarding the model itself. It's also a practical question that has to do with the environment in which a model is used, which means an environment can make a model a better model, right? A model that helps researchers make better scientific contributions. And this means that policies could be designed to create an ideal environment for the kinds of models we want to prefer for policy reasons, right? So non-animal models rather than animal models. I have some ideas in mind. These are preliminary ideas for what could be done, and they're taken from the real world, because some governments and institutions are already trying things in line with this idea of tilting this environment towards non-animal models. One example would be the focus group organ on chip standardization roadmap that was released last year. People who develop and work with organs on chip in this roadmap have laid out the kinds of standards very specifically that their community would still need in order to work together more efficiently. So really, we've got the work cut out for us here. We know how to make organ-on-chip technology more standardized, and that will help it to become more useful to researchers. So if policymakers want, they can allocate resources to this work being done. So that's a very simple kind of measure that we could take. It doesn't have to do anything with putting a cap on animal experimentation licenses or anything like that. It's really about building up standardized databases and vocabulary. Another example would have to be would have to do with financial considerations. When you use a less established non-animal model, if you're the first at your institution, then you'll have to do a lot of labor in addition to your actual scientific experimental work that you won't be compensated for in terms of career benefits. So you'll have to figure out where to get the model, how to use it, and so on. This will not immediately translate into more publications and citations for you. So you're taking a hit for going the extra mile. And a Dutch science funder called ZonMW has recognized this and compensates for this extra labor in the form of top-up funding. So if you make your project in biomedicine animal-free, then they'll give you 50,000 euros on top of your project budget. And that could hire you, a PhD for another year or so, which might then again translate into more publications and citations. So you get something out of it. Of course, you could go beyond these 50,000 to even give non-animal models an edge over animal models if you want. Community support is another foot in the door if policymakers want. If you don't have a community of researchers that use non-animal models or certain types of non-animal models, let's say organoids, then you can try to build communities or at least encourage their growth in a specific place. The Dutch have invented what's called the Helpathon workshop. That's a workshop where different researchers come together. One of them brings a project idea or research question that would require animal experimentation. They discuss and they try to find a version of that initial research idea that doesn't require animal experimentation. So maybe you come in with an animal research idea and you leave with an idea for a project that uses organs on chip. This has the effect of making individual projects less reliant on animal experimentation, which is a good thing, but also it brings people together in different constellations. So this encourages collaboration across different disciplines and creates new networks. Of course, you can also use other funding instruments to bring people together in specific constellations. I noticed this as part of this national research project, national research program. I mean, these 100 people that are funded together also talk to each other. And that's really a very practical way in which you can foster a community. Again, the point is not to just prefer non-animal models over animal models. The idea is to make non-animal models better models because they have an ideal environment in which they can thrive. So that would be one broad kind of policy that I think would be interesting to explore more, tilting the landscape towards innovation in non-animal models. The other question I raised was, how do researchers select scientific purposes? Again, my assumption in the background is scientists want to develop the new best model for the questions and topics they're interested in. So it's essential that we think about how they choose these topics and questions. And there is a field of meta-research or science of science that can give us some guidance on this issue. Scientific topic selection has itself become an object of study in recent years, and one model that appears to be quite well supported by evidence is the one I'm showing you here. This describes an interactive mechanism between an individual scientist and the environment in which they pursue their research. The idea is essentially that a scientist is interested in career benefits, has to be interested in career benefits, because otherwise they won't remain in academia for very long. And they try to get career benefits like a promotion or funding by publishing well. And they start out publishing on certain topics, maybe topics in line with their educational background, and then they see how the environment reacts and they learn from that. The environment that consists, for instance, of the publication landscape, the funding landscape, and the scientific community. And this rewards certain topic selections more than others in terms of how easy it is to get into good journals, how much response you get in terms of citations, because of course a well-cited paper is much more valuable on your CV than a poorly cited one. And it seems to be the case that scientists are fairly good at gravitating towards the topics where they are rewarded by their environment. Not everybody does it at the same pace and in the exactly same way, but generally people do respond to incentives set by the environment, which means, again, there is a foot in the door for policy. If we want, we can try to shape an environment, particularly by funding communities that develop and use specific non-animal models like organs on chip or organoids and cell cultures and computer simulations, so that there is a context of people who exchange about these topics that are generally interested in making progress in those fields. In other words, you can make it rewarding to ask questions that non-animal models can help to answer, incentivizing researchers to seek progress where it doesn't take harm to animals. And I think together, tilting the environment towards innovation of non-animal models and making it rewarding to ask questions that non-animal models can help to answer describes a policy approach that, at least at first sight, seems promising to address the entry side of Lane-Petter's pipeline, to decrease the numbers of new animal models that we get that enter into the pipeline that we then have to replace, without, however, costing a lot in terms of scientific advancements, because we are still developing the best new models. It's just that we've created an environment in which the best new models happen to be non-animal based. I would call this the displacement approach, in contrast to replacement, right? Because it displaces one kind of innovation in models by another. And I think together, replacement and displacement could be a full policy program that can reliably decrease the numbers of animals within the pipeline, and all of that without too much cost to science. So I thank you very much for your attention. If you'd like to know more about what I think on these issues, you can check out my publications. The QR code will lead you to my ORCID page. I can return to this page later if you want, but here are my references in a very small font. And now I really thank you for your attention and look forward to the discussion.